Good morning. My work is on analysis of human cortical progenitor CRISPR knockout screen. These are some of the cortical malformations. And as high as 30% of the malformations that stem from genetic defects cannot be attributed to specific genetic causes. For their prevention and to inform on the non-genetic causes, it is necessary to identify genes and pathways regulating cortical development. This is not an easy task, but with emergence of in vitro systems for cortical development like iPSC-derived human neural progenitor and CRISPR technology, now we can conduct genome-wide CRISPR knockout screens to identify the regulating genes. Livesey Group conducted such screens on iPSC-derived human neural progenitor cells. This is how the screen works. The Cas9 enzyme requires a guide of 20 base pair length RNA to knock out a gene. Let's say, for example, ribosomal gene. This guide is similar to the exonic sequence of this ribosomal gene and binds to the complementary ribosomal gene site in the cell. This is followed by binding of the Cas9 enzyme and cleavage of the site. This break will be repaired by an error-proof system and causes nonsense mutations, which practically kills the gene. We use lentiviral system to deliver these guides, and these get integrated into the cell genome. And because the ribosomal gene is essential to the cell, these cells cannot proliferate when compared to controls. And these differences can be measured by the end of the screen. This is the kind of data generated from the screen. These guides target NF1, these NF2. S stands for the screen and D for the day. I use Toledo data as comparison throughout the project because these 8 screens conducted on neural stem cell lines CB660 and U5 are almost identical to our screens. And these are the guide count differences arising from non-uniform depth of sequencing. Further QC using dimensionality reduction showed that day 2 and day 10 are very similar. And screen 3 is completely different from screen 1 and 2. So these were excluded from further analysis. Variation in day 20 is acceptable. For analysis proper, I use Magic CB2, Bagel2, and EDGAR. These are different in terms of modeling of the data, guide, and gene level statistical tests and output parameters. But more importantly, Bagel uses supervised learning and outputs only depleted genes in this setting. And these are the top 200 depleted and enriched genes identified by these algorithms. Notice that Magic CB2 and Bagel2 agree with each other, and EDGAR is an outlier. There's more agreement between Magic and Bagel2 compared to others. This is also the case with Toledo data, with one exception. Magic and CB2 has highest overlap in U5 dataset. So this correlation can also be observed with the ranks attributed by each algorithm. These are the plots for significantly enriched genes by Magic and EDGAR operating on U5 dataset. These rankings are attributed by EDGAR and Magic for these genes. You can see that EDGAR agrees with Magic's ranking, but the converse is not true. And this testing was also done for all the genes, and significant meaning false discovery rate of less than 10% and p-value of less than 0.05. Base factor of greater than 7 for Bagel. These are Spearman's correlation coefficients for depleted genes. Here the column name indicates the source of significant genes. Magic means that these are correlation numbers for Magic's and other algorithms rankings for Magic's significant genes. You can see that Magic, CB2 and Bagel more or less agree with each other, and EDGAR is an outlier. So to conclude, cross-validation and rank correlation shows that there is more agreement between Magic and Bagel than any other set of algorithms. To understand the validity of each algorithm's results, I compared gene ontology terms obtained from Go analysis of results from each algorithm with reference set of Go terms. This reference set is generated from analysis of expression data, BrainSpan Atlas for developing adult brain, and represent the processes regulating neurodevelopment. This is the comparison between U5 Magic molecular function terms obtained from enriched and depleted genes and reference set of molecular function Go terms. This comparison was done for whole sets of Go terms, molecular function, and biological process subset for all the data sets and all the algorithms, and the percentage of overlap was calculated, which is surprisingly high for EDGAR in general, but Magic and Bagel perform best with Livesey data. And these are the 19 significant genes common to Magic and Bagel2. These are the biological processes which are related to these genes, which can be categorized into 3 sets: protein localization, viral related, and transcription translation. The enrichment and depletion suggest that these genes regulate proliferation, differentiation, and survival. The presence of protein localization in Go analysis suggests these genes might regulate polarity, stemness, and differentiation. This, in conjunction with the fact that most of the identified genes play a role in neurodevelopment, adds to the validity of the screens and usage of these screens to identify genes regulating cortical development. However, the experimental validation of these genes has to be done to see if the performance testing methodology reduces FDR. Validation along with measurement of markers for suggested roles will also dissect the role of these genes in neurodevelopment.